In the previous video, we talked about the idea that if we only use one cycle of the Calvin cycle, by the time we get to G3P, and if we remove the 2G3P that we have produced in order to make glucose, we no longer have any material to be able to recycle RuBP and regenerate it in order to keep this cycle going. So in this video, we're going to look at the solution to how we not only can create our desired glucose molecule, but how we can end up with enough glyceraldehyde 3-phosphate to use in order to regenerate our starting molecule, ribulose 1,5-bisphosphate. And the solution is actually relatively simple. This limitation, the fact that we only have 2 G3P molecules, which is only enough to generate one glucose, is only a problem if we only have one Calvin cycle happening at the same time. But this is ridiculous because the chloroplast is a large organelle, so it logically doesn't make any sense that we would only be having the Calvin cycle happening at once in a single location in the entire organelle. So the question is, how many Calvin cycles do we need in order to be able to make enough G3P to make glucose and still have enough G3P to regenerate RuBP and keep the cycle going indefinitely? Well, we can actually look to the overall photosynthesis reaction for a little bit of a hint. Because if we want to look at the overall reaction, we can simply balance this to give us a hint. We see that there are 6 carbons in glucose, which logically means that we need 6 carbon dioxide molecules, and because glucose follows the CnH2n formula of monosaccharides, that means that we would also need 6 water molecules in order to give us the water that we need in glucose, producing 6 oxygen. Now we know from the second step here that this is the only step in which carbon dioxide is fixed because of the actions of Rubisco, which means that if we want our Calvin cycle to line up with the balancing of photosynthesis overall, we would need to do this reaction 6 times in order to add 6 carbon dioxides to 6 different RuBP molecules, producing 6 pairs of phosphoglyceric acid, and so on. So let's see the results that we would get if we did the Calvin cycle 6 times, just for the sake of argument. Now, if we start at RuBP at the beginning, we can see that RuBP has 1, 2, 3, 4, 5 carbons, and if we did this reaction 6 times, well, then we would have a total of 30 carbons here worth of RuBP, because 6 molecules of RuBP, if it has 5 carbons, is a total of 30 carbons. Now, to that, if we have 6 molecules of CO2, like we see in the overall reaction, we are going to add 6 more carbons from the 6 CO2 molecules when carbon dioxide is fixed by Rubisco. And if we have 6 of this unstable compound that breaks into our 3-PGA molecule, well, now we have 6 times this compound, which has 6 carbons, which is a total of 36 carbons, and that makes sense because RuBP has 30, and if we add 6 to that, we get 36 carbons, which matches exactly as if we were to take this compound and have 6 copies of it. Then, this compound here breaks apart because of the unstable negative charge, and again, if we have 6 Calvin cycles happening here, we have 2 3-carbon compounds because 3-PGA has 1, 2, 3, and a second one has 1, 2, 3, so the number of our carbons hasn't changed at all. And again, because of this stage here, because we are phosphorylating this oxygen here and this oxygen here, we aren't changing the number of carbons, so we would still, if we're doing this reaction 6 times, have a total of 36 carbons here. The only thing that would change is if we're multiplying the number of reactions that we're doing, we also need to do the same with the ATP, meaning that for 6 Calvin cycles, we would need 12 ATP in total to phosphorylate every one of the 3 PGA molecules, because again, we would have 12 in this case here. And the most important step is here, because if we have 36 carbons worth of G3P molecules at this time, this is where we're going to take some, but not all, of our G3P and separate it, put it aside as an investment, if you will, in order to make glucose. Now, in order to make one glucose molecule, which is C6H12O6, we need 6 carbons. And if we were to take, because glyceraldehyde 3-phosphate is a 6-carbon molecule here, if we were to take 6 carbons away from the 36 carbons worth of glyceraldehyde 3-phosphate, we would have a total of 30 carbons left over. Now, why is this number here significant? Well, remember, the whole reason why we need to do 6 Calvin cycles worth of reactions is because we need enough carbons to regenerate RuBP. Now, if we pay attention, if we say that all of the G3P that we have left over after we've put aside some to make glucose is 30 carbons worth, that number should seem familiar, because 6 RuBP molecules, our starting compound, have exactly 30 carbons. So for the first time, we can see that if we do Calvin cycle 6 times, not only would we generate enough G3P to generate one molecule of glucose, but the leftover carbons, or from the leftover G3P molecules that we would have, would be enough to regenerate all 6 of the RuBP molecules that we originally needed to start this chemical reaction. Now, in terms of understanding this, the more connections that you can make, the more it will make sense. So, we can find that this number 30 here is relatively significant as well, because if we take the lowest common multiple between the number of carbons in RuBP, which is 5, and 2 G3P molecules, which have 6 carbons, the lowest common multiple between 5 and 6 is 30, which is exactly the number of carbons that we need to make 6 molecules of RuBP, and is exactly the number of carbons that we have if we take 6 cycles worth of G3P, remove enough G3P to make glucose, and we see that we have exactly 30 carbons worth of G3P left over. Now, a different way that we can look at this is by actually counting the number of G3P molecules that we have. So if we did 6 Calvin cycles worth, we would have 12 G3P molecules. Now, we're going to subtract 2 G3P molecules from this because we need those to make glucose, meaning that we will have 10 G3P molecules left over, which again have a total of 30 carbons. And those 10 G3P molecules are exactly enough to reform 6 RuBP molecules, because if we have 10 G3P molecules, that contains 30 carbon in total, which is exactly enough to reform the 6 RuBP that we began this reaction with. So the conclusion to this is that it takes 6 Calvin cycles happening concurrently in order to regenerate ribulose 1,5-bisphosphate, our starting compound, while simultaneously having enough G3P to produce one glucose molecule. And of course, we need to make this change as well for the amount of ATP we need and the amount of NADPH that we would need, because all of these are reactants for a Calvin cycle as well. So the only thing that remains to discuss is how we can actually get our glucose 6 or our glucose 3 phosphate back to RuBP. So as we recall, 6 cycles of producing 2 molecules of G3P will result in 36 carbons. Therefore, since it takes 6 carbons in order to make glucose, we are going to remove 6 carbons, which is equal to 2 G3P molecules away in order to make glucose, and then we would have left over from this 30 carbons worth of G3P, also called 10 individual G3P molecules, which is exactly enough to produce 6 RuBP molecules, because if RuBP has 5 carbons, 6 times 5 is a total of 30 carbons required. Now, the only problem actually doesn't have anything to do with carbon now. The problem is phosphate now, because we can see that glyceraldehyde 3-phosphate only has 1 phosphate attached to it, but we can see RuBP has 2. So therefore, we also need ATP, 1 ATP for each glyceraldehyde 3-phosphate, or rather 1 ATP for each RuBP that we are making. So we need ATP to add a second phosphate to each molecule, meaning that we need, because we are making 6 RuBP molecules at the end, we need a total of 6 ATP in order to add the second phosphate here. And as a consequence of this, as a consequence of doing the Calvin cycle 6 times concurrently, we should be able to get all 6 RuBP molecules regenerated because of the exact balancing that this gives us within the amount of G3P produced, even subtracting the G3P that is needed to form glucose once again. This concludes our video series on the Calvin cycle. To review, make sure that if you read a shorthanded diagram of the Calvin cycle like this, make sure that you can understand the balancing of carbon and keep track that the amount of carbon that we start with is the same as the amount of carbon that we regenerate at the end, as long as we have 6 Calvin cycles operating concurrently.